We are starting the chapter first of the class 12th, that is sexual reproduction in flowering plants. ठीक है, तो चलो let's proceed further. Are we not lucky that plants reproduce sexually? The myriads of flowers that we enjoy gazing at, the scents and the perfume that we son over, the rich colors that attract us are all there as an aid to sexual reproduction. Flowers do not exist only for us to be used for our own selfishness. All flowering plants show sexual reproduction. क्या है? ट्रू स्टेटमेंट है, ट्रू है बिल्कुल, एनसीईआरटी में दिया हुआ है. ऑल फ्लावरिंग प्लांट्स शोज सेक्सुअल रिप्रोडक्शन. A look at the diversity of structure of the inflorescence flowers and floral parts show an amazing range of adaptation to ensure formation of the end products of sexual reproduction, the fruit and seeds. In this chapter, let us understand the morphology, structure and the process of sexual reproduction in flowering plants, that is angiosperms. ठीक है, जो फास्ट पढ़ता हूँ वो काम की नहीं होती है. लाइन्स मीन्स कुछ काम में नहीं आती है, इसलिए मैं फास्ट पढ़ता हूँ, और बाकी जहाँ काम में आती है वो लाइन में डबल और स्लो कर देता हूँ. ठीक है. नेक्स्ट, फ्लावर, ए फैसिनेटिंग ऑर्गन ऑफ एंजियोस्पर्म. ह्यूमन बीइंग हैव हैड एन इंटीमेट रिलेशनशिप विद फ्लावर since time immemorial. Flowers are subject of aesthetic, ornamental, social, religious and cultural value. They have always been used as symbol for conveying important human feelings such as love, affection, happiness, grief, mourning, etc. List at least 5 flowers of ornamental value that are commonly cultivated at homes and in gardens. Find out the names of 5 flowers that are used in social and cultural celebration in your family. Have you heard of floriculture? What does it refer to? To a biologist, flowers are morphological and embryological marvels and the site of sexual reproduction. In earlier classes, you have read the various parts of the flower. Figure will help you recall the parts of the typical flower. Can you name the 2 parts in a flower in which the 2 most important units of sexual reproduction develop? दे सकते हो क्या नाम? Can you name the 2 parts in the flower in which the 2 most important units of sexual reproduction develops? कौन से वो पार्ट है? एंड्रोशियम और गाइनोशियम. ठीक है. The next is pre-fertilization. That is structure and event. We are going to read pre-fertilization. Much before the actual flower is seen on a plant, the decision that the plant is going to flower has taken place. Several hormones and structural changes are initiated, which lead to a differentiation and further development of the floral primordium. Inflorescences are formed which bear the floral buds and then the flowers. In the flower, the male and the female reproductive structure, the androecium and the gynoecium, differentiate and develop. You would recollect that the androecium consists of a halls of stamens representing the male reproductive organ and the gynoecium represents the female reproductive organ. We are going to read stamen, that is microsporangium and pollen grain. In the figure shows the 2 parts of a typical stamen, the long and slender stalk called the filament. Long and slender stalk is called the filament. ठीक है. And the terminal generally bilobed structure called the anther. And the terminal generally bilobed structure called the anther. The proximal end of the filament is attached to the thalamus or the petal of the flower. Proximal end of filament is attached to the thalamus or the petal of the flower. The number and length of the stamen are variable in flowers of different species and the flower of different species. If you were to collect stamens each from 10 flowers, each from different species, and arrange them on a slide, you would be able to appreciate a large variation in size seen in nature. Careful observation of each stamen under a dissecting microscope and making neat diagram would elucidate the range in shape and arrangement of anthers in different flowers. A typical angiosperm anther is bilobed, with each lobe having two theca. That is, they are dithecous. Often a longitudinal groove runs lengthwise, separating the theca. Let us understand the various types of tissues and their organization in the transverse section of an anther. The bilobed nature of an anther is very distinct in the transverse section of the anther. The anther is a four-sided, that is tetragonal, structure consisting of four microsporangia located at the corners, two in each lobe. Anther is a four-sided tetragonal structure consisting of four microsporangia located at the corners, two in each lobe. The microsporangia develop further and become pollen sacs. Microsporangia develop further and become pollen sacs. They extend longitudinally all through the length of an anther and are packed with pollen grains. Structure of microsporangium. ठीक है, क्या पढ़ रहे हैं हम? Structure of microsporangium. In a transverse section, a typical microsporangium appears near circular in outline. It is generally surrounded by four wall layers: the epidermis, endothecium, middle layer and the tapetum. ठीक है, किस किस से सराउंडेड होता है? फोर लेयर होती है, एपिडर्मिस, एंडोथीशियम, मिडिल लेयर एंड द टैपेटम. ठीक है, क्लियर है? हो गया माइंड में सेट? ठीक है, क्लियर? द आउटर थ्री वॉल्स परफॉर्म the outer three wall layer perform the function of protection and helps in dehiscence of anther to release the pollen. कौन सी थ्री आउटर? एपिडर्मिस, एंडोथीशियम और मिडिल लेयर. The outer three wall layers perform the function of protection and help in dehiscence of anther to release the pollen. The innermost wall layer is the tapetum. It nourishes the developing pollen grain. Cells of the tapetum possesses dense cytoplasm and generally have more than one nucleus. यहाँ पे कंफ्यूज करने के लिए जनरली वन न्यूक्लियस कर देगा, बट करेक्ट है, जनरली हैव मोर देन वन न्यूक्लियस, ये करेक्ट है. कैन यू थिंक ऑफ हाउ टैपेटल सेल्स कुड बिकम बाइन्यूक्लिएट? ये आप ही सोचिए. ठीक है. When the anther is young, a group of compactly arranged homogeneous cells called the sporogenous tissues occupies the center of each microsporangium. When the anther is young, a group of compactly arranged homogeneous cells called the sporogenous tissue occupies the center of each microsporangium. Microsporogenesis, as the anther develops. Sporogenesis. As the anther develops, the cells of the sporogenous tissue undergo meiotic divisions to form microspore tetrads. Microsporogenesis पढ़ रहे हैं. As the anther develops, the cells of the sporogenous tissue undergo meiotic divisions to form microspore tetrads. What would be the ploidy of the cells of the tetrads? Answer me. What would be the ploidy of the cells of the tetrad? That is haploid. Tetrad जो होते हैं वो haploid होते हैं. This is because tetrads are formed through meiosis, which produces cells with half the ploidy of the original cell. ठीक है? Understood? So we are proceeding further. ठीक है, डायग्राम दिया हुआ है, देख लेना. As each cell of the sporogenous tissue is capable of giving rise to a microspore tetrad. Each one is a potential pollen or microspore mother cell. The process of formation of microspore from a pollen mother cell. The process of formation of microspores from pollen mother cells PMC through meiosis is called microsporogenesis. The microspores as they are formed are arranged in a cluster of 4 cells. The microspore tetrad. The microspores as they are formed are arranged in a cluster of 4 cells. The microspore tetrad. As the anther mature and dehydrate, the microspore dissociate from each other and develop into pollen grains. As the anther mature and dehydrate, the microspore dissociate from each other and develop into pollen grains. Inside each microsporangium, several thousands of microspores or pollen grains are formed that are released with the dehiscence of the anther. Next is pollen grain. Dehiscence का मतलब क्या होता है यार? Splitting of a bone or a plant structure. हिंदी में जिसको कहते हैं स्फुटन, मतलब फूटना. Pollen grain. The pollen grain represents the male gametophytes. If you touch the opened anther of hibiscus or any other flower, you would find deposition of yellowish powdery pollen grain on your fingers. Sprinkle these grains on a drop of water taken on a glass slide and observe under the microscope. You will really be amazed at the variety of architecture, size, shape, color, design seen on the pollen grain from different species. ठीक है, यहाँ पे नया डायग्राम दिया हुआ है NCERT में. Scanning electron micrographs of a few pollen grain, जो पहले वाली NCERT में नहीं दिया हुआ. ठीक है, डायग्राम ऐड हुआ है. एक enlarged view of a pollen grain tetrad का और एक stages of microspore maturing into a pollen grain. ठीक है. So the next, pollen grains are generally spherical, measuring about 25 to 50 micrometers in diameter. Pollen grain are generally spherical, measuring about 25 to 50. 25 का double 50 micrometers in diameter. It has prominent 2-layered walls. The hard outer layer called the axine. Is made up of sporopollenin, which is one of the most resistant organic material known. बहुत क्वेश्चन आते हैं यहाँ से. It can withstand high temperature and strong acids and alkalis. No enzyme that degrade sporopollenin is so far known. अभी तक ऐसा एंजाइम नहीं आया जो इसकी वॉट लगा दे. Pollen grain axine has prominent apertures called germ pores where sporopollenin is absent. यहाँ पे नहीं होता है sporopollenin. कहाँ पे? germ pore पे. ठीक है. Pollen grain are well preserved as fossils because of the presence of sporopollenin. The axine exhibit a fascinating array of patterns and designs. Why do you think the axine should be hard? What is the function of germ pore? बताओ function of germ pore. ठीक है, बताते हैं अभी. Function of germ pore is to initiate the pollen tube and release male gamete during fertilization. ठीक है, यह होता है function germ pore का. The inner wall of the pollen grain is called the intine. It is a thin, यहाँ पे thick कर सकता है but correct है thin. It is a thin and continuous layer made up of cellulose and pectin. किससे बनी होती है? Cellulose और pectins. The cytoplasm of pollen grain is surrounded by a plasma membrane. किससे surrounded होता है? Plasma membrane. When the pollen grain is matured, it contains 2 cells, the vegetative cell and the generative cell. The vegetative cell is bigger, has abundant food reserve and a large irregularly shaped nucleus. Irregularly shaped nucleus. The vegetative cell is bigger, has abundant food reserve and a large irregularly shaped nucleus. The generative cell is small. Generative cell small होता है, ठीक है, and floats in the cytoplasm of the vegetative cell. Float करता है cytoplasm vegetative cell के. It is spindle shaped with dense cytoplasm and a nucleus. In over 60% of angiosperm, pollen grain are shed at this two-celled stage. ठीक है, कितनी 60%. वापस रिपीट कर रहा हूँ, in 60% of angiosperm, pollen grain are shed at this two-celled stage. कौन से स्टेज पे? वेजिटेटिव सेल एंड जनरेटिव सेल वाली स्टेज पर, ठीक है. In the remaining species, the generative cell divides methodically to give rise to the two male gametes before pollen grain are shed. That is three-celled stage. Remaining species, the generative cell, कौन सा सेल? जनरेटिव, जनरेट करता है, ठीक है. जनरेटिव सेल डिवाइड्स मिटोटिकली, यहाँ की जगह चेंज कर सकता है बट करेक्ट है मिटोटिकली, माइटोटिकली, टू गिव राइज टू द टू मेल गैमेट्स बिफोर pollen grain are shed. That is three-celled stage, ठीक है. Pollen grain of many species cause severe allergies and bronchial afflictions in some people, often leading to chronic respiratory disorder, asthma, bronchitis, etc. It may be mentioned that Parthenium or carrot grass that came into India as a contaminant with imported wheat has become ubiquitous in occurrence and cause pollen allergy. कौन सा? Parthenium or carrot grass come into India as contaminant with imported wheat has become ubiquitous in occurrence and causes pollen allergy, ठीक है. Pollen grain are rich in nutrients. Pollen grains are rich in nutrients. It has become a fashion in recent years to use pollen tablets as food supplements. In Western countries, a large number of pollen product in the form of tablets and syrups are available in the market. Pollen consumption has been claimed to increase the performance of athletes and racehorses. किसका consumption? Pollen consumption has been claimed to increase the performance of athletes and racehorses, ठीक है. डायग्राम दिया हुआ है. बी pollen from England, 30 टेबलेट दी हुई है उसमें, ठीक है. Natural bee collected pollen in tiny golden pods. A natural food from honeybees. Net weight 45 Aussie. डायग्राम में दिया हुआ है, ठीक है. When once they are shed, pollen grain have to land on the stigma before they lose viability. If they have to bring about fertilization, ठीक है. फर्टिलाइजेशन करना होगा तो क्या करना होगा? Pollen grain have to land on the stigma before they lose viability. How long do you think the pollen grain retain viability? The period for which pollen grain remain viable is highly variable and to some extent depend on the prevailing temperature and humidity. In some cereals such as rice and wheat, pollen grain lose viability within 30 minutes of their release. राइस एंड व्हीट में 30 मिनट में वाइबिलिटी लॉस हो जाती है. राइस और अपन ऐसे याद रख सकते हैं, राइस और व्हीट सब बना मीन्स रोटियाँ जल्दी खा लेते हैं 30 मिनट में, ठीक है. भैया बकवास ट्रिक बताइए मैंने बट काम किए, ठीक है. इंस्टेंट आई थी वो बता दी, मेरे को भी नहीं पता था, ठीक है. इन सम मेंबर्स ऑफ रोसेशिया, लेग्यूमिनेशिया and Solanaceae, they maintain viability for months. 4 month नहीं 4 months मतलब महीनों तक, ठीक है. कौन से? इन सब मेंबर्स ऑफ रोसाशिया, लेग्यूमिनेशिया एंड सोलेनेशिया, RLS, they maintain viability for months. You may have heard of storing semen, sperms of many animals including humans for artificial insemination. It is possible to store pollen grain of a large number of species for years in liquid nitrogen, that is -196 डिग्री सेल्सियस। यहाँ पे ऐसा भी ऑप्शन दे सकते हैं 195, 96, 97, 98, बट करेक्ट है -196 डिग्री सेल्सियस। और ऑप्शन ऐसे आएंगे, माइनस भी देगा एक और एक प्लस भी देगा, -196 और +196। तो आपको करेक्ट करना होगा -196। इस लेवल पे बना रहा हूँ मैं क्वेश्चन जो डायरेक्ट आते हैं, ठीक है, और बहुत ट्रिकी बनाया है। इन लिक्विड नाइट्रोजन -196 डिग्री सेल्सियस, ठीक है। सच स्टोर्ड pollen कैन बी यूज्ड एज pollen banks similar to seed banks in crop breeding programs. It is possible to store pollen grain of large number of species for years in liquid nitrogen at -196 degree Celsius. ठीक है। Next is the pistil, megasporangium ovule and embryo sac. ठीक है। The gynoecium represent the female reproductive part of the flower. क्या रिप्रेजेंट करता है? जाइनोशियम फीमेल रिप्रोडक्टिव पार्ट ऑफ द फ्लावर। द जाइनोशियम में कंसिस्ट ऑफ ए सिंगल पिस्टल दैट इज मोनोकार्पेलरी और में हैव मोर देन वन पिस्टल मल्टी कार्पेलरी, ठीक है। when there are more than one the pistil may be fused together syncarpus they are called, or may be free मतलब apocarpus. Each pistil has 3 parts, stigma, style and ovary. क्या क्या होते हैं 3 पार्ट? The stigma, style and ovary. The stigma serves as a landing platform for pollen grain. Stigma landing platform है, ठीक है। कौन है लैंडिंग प्लेटफॉर्म? स्टिग्मा, ठीक है। The style is the elongated slender part beneath the stigma. स्टिग्मा के नीचे वाला। The basal bulged part of the pistil is the ovary. Inside the ovary is the ovarian cavity, that is locule. The placenta is located inside the ovarian cavity. Ovarian cavity के अंदर होता है placenta. Recall the definition and types of placentation that you have studied in the 11th class. ठीक है। Arising from the placenta are the megasporangia, commonly called ovules. यहाँ पर डायग्राम दिया हुआ है, डाइसेक्टेड फ्लावर ऑफ हिबिस्कस शोविंग पिस्टल अदर फ्लोरल पार्ट हैव बीन रिमूव्ड। ये डायग्राम दिया हुआ है, उसमें स्टिग्मा, स्टाइल, ओवरी बताया हुआ है और थैलेमस बताया हुआ है। सेकंड दिया हुआ है मल्टी कार्पेलरी सिनकार्पस पिस्टल ऑफ पेपावर, मल्टी कार्पेलरी सिनकार्पस पिस्टल ऑफ पेपावर। और थर्ड दिया हुआ है मल्टी कार्पेलरी एपोकार्पस जाइनोशियम ऑफ माइशेलिया। और डायग्रामेटिक व्यू ऑफ टिपिकल एनाट्रोपस ओव्यूल का दिया हुआ है, जिसमें दिया हुआ है फ्यूनिकल, माइक्रोपाइल, पाइलर पोल, इंटीग्यूमेंट, न्यूसेलस, ये दिए हुए डायग्राम, ठीक है। Arising from the placenta are the megasporangia, commonly called ovules. The number of ovules in an ovary may be 1, that is in wheat, paddy, mango. Wheat और paddy और mango में होते हैं ovule. The number of ovules in an ovary may be 1. Ovules ovary के अंदर 1 होते हैं, वो होते हैं wheat में, paddy में और mango में। Too many होते हैं पपाया, वाटरमेलन और ऑर्किड्स। पपाया, वाटरमेलन और ऑर्किड्स, पपाया, वाटरमेलन और ऑर्किड्स, ठीक है। हो गया सेट, पपाया, वाटरमेलन और ऑर्किड्स, क्लियर। The megasporangium ovule. Let us familiarize ourselves with the structure of a typical angiosperm ovule. The ovule is a small structure attached to the placenta by a means of a stalk called funicle. The body of the ovule fuses with the funicle in the rind called hilum. Thus, hilum represents the junction between ovule and funicle. किसके बीच का जंक्शन है? ओव्यूल और फ्यूनिकल के बीच का। hilum represents the junction between the ovule and funicle. Clear? जंक्शन है ओव्यूल और फ्यूनिकल के बीच का hilum. Each ovule has 1 or 2 protective envelopes called integuments. Integuments encircle the nucellus except at the tip where a small opening called the micropyle is organized. Integuments encircle the nucellus except at the tip where a small opening called the micropyle is organized. ठीक है. Opposite the micropylar end is a chalaza. Opposite the micropylar end is the chalaza, representing the basal part of the ovule. Chalaza क्या represent करता है? Basal part of the ovule. Opposite होता है micropylar end के. ठीक है. Enclosed within the integuments is a mass of cells called the nucellus. Cells of the nucellus have abundant reserve materials. ठीक है. Corrected reserve food material. Located in the nucellus is the embryo sac or female gametophyte. An ovule generally has a single embryo sac formed from a megaspore. An ovule generally has a single embryo sac formed from a megaspore. Next is megasporogenesis. The process of formation of megaspores from the megaspore mother cell is called megasporogenesis. Ovules generally differentiate a single megaspore mother cell, that is MMC, in the micropylar rind of the nucellus. ठीक है. Ovules generally differentiate a single megaspore mother cell MMC in the micropylar rind of the nucellus. ठीक है. Megasporogenesis. The process of formation of megaspore from the megaspore mother cell is called megasporogenesis. ठीक है. डायग्राम दिया हुआ है यहाँ पे माइक्रोपाइलर एंड, न्यूसेलस, मेगास्पोर मदर सेल. ठीक है. यहाँ पे सिनर्जिड बताई हुई है, एग बताया हुआ है, सेंट्रल सेल, टू पोलर न्यूक्लियाई, एंटीपोडल सेल. पार्ट ऑफ द ओव्यूल शोइंग ए लार्ज मेगास्पोर मदर सेल, ए डायड एंड ए टेट्राएड ऑफ मेगास्पोर्स. ठीक है. और यहाँ पे दिया हुआ है डायग्राम 2, 4 and 8 न्यूक्लियर स्टेजेस ऑफ एम्ब्रियो एंड एम्ब्रियो सेक. ठीक है. और किसका डायग्राम दिया हुआ है? एम्ब्रियो सेक का भी दिया हुआ है, जहाँ पे ऊपर है चालेजल एंड, चला जाओ ऊपर एंड पे. ठीक है. उसमें है एंटीपोडल्स, बीच में है पोलर न्यूक्लियाई, सेंट्रल सेल है, और नीचे है माइक्रोपाइलर एंड, फिलिफॉर्म अपैरेटस दिया हुआ है, सिनर्जी दी हुई है और एक दिया हुआ है. ठीक है. It is a large cell containing dense cytoplasm and a prominent nucleus. ठीक है. Ovule generally differentiate a single megaspore mother cell MMC in the micropylar rind of the nucellus. It is a large cell containing dense cytoplasm and a prominent nucleus. The microspore mother cell undergo meiotic division. What is the importance of the MMC, that is microspore mother cell undergoing meiosis? Meiosis results in the production of 4 megaspores. कितने? 4 megaspores. Next is female gametophyte. ठीक है. क्या पढ़ रहे हैं नेक्स्ट? फीमेल gametophyte. In a majority of flowering plants, one of the megaspore is a functional, while the other 3 degenerate. एक ही होता है functional, बाकी other 3 degenerate. Only the functional megaspore develop into the female gametophyte. Embryo sac. ठीक है. जिसे female gametophyte को embryo sac कहते हैं. This method of embryo sac formation from a single megaspore is termed monosporic development. Monosporic मतलब एक से development. ठीक है. What will be the ploidy of the cells of the nucellus, microspore mother cell, the functional megaspore and the female gametophyte? सुनो ध्यान से. ठीक है. न्यूसेलस होता है 2n, माइक्रोस्पोर मदर सेल होता है 2n, फंक्शनल मेगास्पोर होता है n, दैट इज हैप्लॉइड, और फीमेल गैमेटोफाइट होता है n, दैट इज हैप्लॉइड. न्यूसेलस भी डिप्लॉइड है, मीन्स 2n, माइक्रोस्पोर मदर सेल भी 2n है, दैट इज डिप्लॉइड. फंक्शनल मेगास्पोर n और फीमेल गैमेटोफाइट भी n है. ठीक है. क्लियर है क्वेश्चन? आ सकता है पक्का, ठीक है। रात के बज चुके हैं 1:40। Let us study about the formation of the embryo sac in detail. The nucleus of the functional megaspore divide mitotically to form two nuclei, which move to the opposite pole, forming the two-nucleate embryo sac. Two or more sequential mitotic nuclear division result in the formation of the 4-nucleate and later the 8-nucleate stages of the embryo sac. It is of interest to note that this mitotic division are strictly free nuclear. That is, nuclear division are not followed immediately by cell wall formation. Nuclear division are not followed immediately by cell wall formation. ठीक है, understood? After the 8-nucleate stage, cell walls are laid down. After the 8-nucleate stage, cell walls are laid down, leading to the organization of the typical female gametophyte or embryo sac. Leading to the organization of the typical female gametophyte or embryo sac. Observe the distribution of the cells inside the embryo sac. 6 of the 8 nuclei are surrounded by cell walls. 6 of the 8 nuclei are surrounded by the cell walls and organized into cells. The remaining 2 nuclei, called polar nuclei, are situated below the egg apparatus. किसके नीचे? Below the egg apparatus in the large central cell. There is a characteristic distribution of the cells within the embryo sac. 3 cells are grouped together at the micropylar end and constitute the egg apparatus. The egg apparatus in turn consists of 2 synergids and 1 egg cell. The egg apparatus in turn consists of 2 synergids and 1 egg cell. Egg cell क्या से कंसिस्ट होता है? 2 synergid और 1 egg cell से, ठीक है। The synergid have special cellular thickenings at the micropylar tip called filiform apparatus, which play an important role in guiding the pollen tubes into the synergid. 3 cells are at the chalazal end and are called the antipodal CA से याद रख सकते हो, C से chalazal end and are called antipodal. The large central cell as mentioned earlier has 2 polar nuclei. Thus, a typical angiosperm embryo sac at maturity through is 8 nucleate is 7 cell. A typical embryo sac at maturity through 8 nucleate is 7 cell. ये क्वेश्चन पूछा हुआ है, ठीक है, PYQ. Next is pollination, a very hot topic. In the preceding sections, you have learned that the male and the female gametes in flowering plants are produced in the pollen grain and embryo sac respectively. Male कहाँ पे होता है? Pollen grain में, और female embryo sac में. As both types of gametes are non-motile, they have to be brought together to fertilization to occur. For the fertilization to occur. How is this achieved? Pollination is the mechanism to achieve this objective. Transfer of pollen grains shed from the anther to the stigma of a pistil is termed as pollination. Flowering plants have evolved an amazing array of adaptations to achieve pollination. They make use of external agents to achieve pollination. Can you list the possible external agents? Kinds of pollination. Depending on the source of pollen, pollination can be divided into 3 types. कितने types? 3 types. First is autogamy. In this type, pollination is achieved within the same flower. Transfer of pollen grain from the anther to the stigma of the same flower. In a normal flower, which opens and exposes the anther and the stigma, complete autogamy is rather rare. In a normal flower which opens and exposes the anther and the stigma, complete autogamy is rather rare. Autogamy in such flowers require synchrony in pollen release and stigma receptivity, and also the anther and the stigma should lie close to each other so that the self-pollination can occur. Autogamy in such flowers require synchrony in pollen release and stigma receptivity, and also the anther and the stigma should lie close to each other so that the self-pollination can occur. Some plants such as Viola, that is common pansy, oxalis and commelina produce two types of flowers. कौन some plants such as Viola, common pansy जिसे कहते हैं, oxalis और commelina, two types of flower produce करते हैं. First is chasmogamous flowers which are similar to flowers of other species with exposed anthers and stigma, and clistygamous flower which do not open at all. In such flowers, the anther and the stigma lies close to each other. When the anther dehisces in the flower buds, pollen grains come in contact with the stigma to effect pollination. When the anther dehisces in the flower buds, pollen grain come in contact with the stigma to effect pollination. Thus, clistygamous flowers are invariably autogamous, as there is no chance of cross-pollination landing on the stigma. कोई चांस ही नहीं क्रॉस पॉलिनेशन करना. Clistygamous flowers produce assured seed set even in the absence of pollinator. Pollinator की absence में भी क्या होता है? Assured seed set. Clistygamous flowers produce assured seed set even in the absence of pollinators. Do you think that cleistogamy is advantageous or disadvantageous to the plant, and why? चलो मैं बताता हूँ आंसर, सुनना. Cleistogamy is generally considered advantageous to a plant because it allows for guaranteed seed production. It allows for guaranteed seed production even in the absence of pollinator, but it is also disadvantageous due to lack of genetic variation caused by self-pollination. Clear है? ठीक है. Next is geitonogamy. Transfer of pollen grain from anther to the stigma of another flower of the same plant. Another flower of the same plant. Although geitonogamy is functionally cross-pollination involving a pollinating agent, genetically it is similar to autogamy since the pollen grain come from the same plant. It is similar to the autogamy since the pollen grain come from the same plant. Next is xenogamy. Transfer of pollen grain from anther to the stigma of a different plant. This is the only type of pollination which during pollination brings genetically different types of pollen grain to the stigma. Genetically different types of pollen grain to the stigma. Next is agents of pollination. Plants uses two abiotic. Abiotic में आते हैं wind and water, and one biotic में आते हैं animals. Plants uses two abiotic में आते हैं wind and water, biotic में आते हैं animals. Wind और water आते हैं abiotic में, and biotic animals agents to achieve pollination. Majority of the plants uses biotic agent for pollination. Only a small proportion of plants uses abiotic agents. Pollen grains coming in contact with the stigma is a chance factor in both wind and water pollination. To compensate for these uncertainties and associated loss of pollen grains, the flowers produce enormous amount of pollen when compared to the number of ovules available for pollination. Pollination by wind is more common amongst abiotic pollination. वो लाइन एक बार और पढ़ता हूँ. टू compensate for the uncertainties and associated loss of pollen grain, the flower produce enormous amount of pollen when compared to the number of ovules available for pollination. Pollination by wind is more common amongst the abiotic pollinations. Wind pollination also requires that the pollen grain are light and non-sticky so that they can be transported in wind currents. ठीक है. Pollination by wind is more common amongst abiotic pollinators. Among abiotic pollination में wind common. ठीक है. Wind is more common. Wind pollination also require that pollen grain are light and non-sticky. Non-sticky जगह sticky कर सकता है but correct है non-sticky so that they can be transported in wind currents. They often possess well-exposed stamens so that the pollens are easily dispersed into the wind currents, and large, often feathery stigma to easily trap airborne pollen grains. Wind-pollinated flowers often have a single ovule in each ovary and numerous flowers packed into an inflorescence. Wind-pollinated flowers often have a single ovule in each ovary and numerous flowers packed into an inflorescence. A familiar example is the cornucopia. The tassels you see are nothing but the stigma and the style which wave in the wind to trap pollen grains. क्या है वो? स्टिग्मा और स्टाइल है जो हवा में लहराते हैं pollen grain को ट्रैप करने के लिए. Wind pollination is quite common in grasses. Important line है. Wind pollination is quite common in grasses. Understood? Pollination by water is quite rare in flowering plants and is limited to about 30 genera, mostly monocotyledons. यहाँ पे चेंज करके mostly dicotyledons कर सकता है but correct है mostly monocotyledons correct है. As against this, you would recall that water is a regular mode of transport for the amalgamates amongst the lower plant groups such as algae, bryophyte, pteridophyte. It is believed, particularly for some bryophytes and pteridophytes, that their distribution is limited because of the need for water, because for the need for water for the transport of amalgamate and fertilization. It is believed, particularly for some bryophytes and pteridophytes, that their distribution is limited because of the need of the water for the transport of amalgamates and fertilization. Clear? Some examples of water pollinated plants are Vallisneria and Hydrilla, which grow in fresh water, and several marine sea grasses such as Justera. फ्रेश वाटर का एग्जाम्पल है वैलिसिनेरिया और हाइड्रिला. मराइन का है सी ग्रासेस सच एज जुस्टेरा. नॉट ऑल एक्वेटिक प्लांट्स यूजेस वाटर फॉर पॉलिनेशन. करेक्ट स्टेटमेंट है. ठीक है. नॉट ऑल एक्वेटिक प्लांट्स यूजेस वाटर फॉर पॉलिनेशन. इन अ मेजॉरिटी ऑफ एक्वेटिक प्लांट्स सच एज वाटर हायासिंथ एंड वाटर लिली, the flower emerges above the level of water and are pollinated by insect or wind, as in most of the land plants, and are pollinated by insect or wind, as in most of the land plants. In Vallisneria, the female flower reaches the surface of the water. In Vallisneria, the female flower reaches the surface of water by the long stalk, and the male flower or pollen grain are released on the surface of water. They are carried passively by water current. चेंज करके एक्टिवली कर सकता है यहाँ पे बट करेक्ट है. They are carried passively by water currents. Some of them eventually reach the female flowers and the stigma. Some of them eventually reach the female flowers and the stigma. Understood? In another group of water pollinated plants such as sea grasses, female flower remain submerged in water and the pollen grain are released inside the water. क्या होता है? Female flower remains submerged in water and the pollen grain are released inside the water. वाटर के अंदर pollen grain रिलीज कर दिए जाते हैं. ठीक है. So let's proceed further. Pollen grain in many such species are long, ribbon-like, and they are carried passively inside the water. Some of them reach the stigma and achieve pollination. कैसे carried out होते हैं? Passively inside the water. Some of them reach the stigma and achieve pollination. In most of the water-pollinated species, pollen grain are protected from wetting by a mucilaginous covering. किससे protected होते हैं? Mucilaginous covering. Both wind and water-pollinated flowers are not very colorful and do not produce nectar. Are not very colorful and do not produce nectar. कौन से? Wind and water से pollinated. What would be the reason for this? Can you please tell me the reason? What would be the reason for that? Majority of the flowering plants use a range of animals as pollinating agents. Bees, butterflies, flies, beetles, wasps, ants, moths, bird में sunbird and hummingbird, and bats are the common pollinating agents. Again I am repeating it. Bees, butterflies, flies, beetles, wasps, ants, moth, birds में sunbirds and hummingbirds, and bats are the common pollinating agent. ठीक है? Clear? Among the animals, insects, particularly bees, are the dominant biotic pollinating agent. कौन होते हैं dominant? Particularly bees are the dominant biotic pollinating agents among the animals. Among the animal insects, particularly bees are the dominant. Understood? Insect और animals में bees dominate होते हैं biotic pollinating agent. Even larger animals such as some primates में कौन आता है? Lemurs. Arboreal, that is tree dwelling rodents, or even reptiles, jackdaw lizard and garden lizard, have also been reported as pollinators in some species. Even larger animals such as some primates, lemurs, arboreal, that is tree dwelling rodents, or even reptiles, that is jackdaw lizard and garden lizard, have also been reported as pollinators in some species. Often flowers of animals pollinated plants are specifically adapted for a particular species of animals. Often flowers of animal pollinated plants are specifically adapted for a particular species of animals. आँखें बंद करके सुना करो, ठीक है? और स्लोली जहाँ पे पढ़ता हूँ वहाँ पे रिलैक्सेशन टाइम देता हूँ ताकि लाइन सेट हो जाए. इसलिए पॉडकास्ट आपको बहुत हेल्प करेगा. ठीक है, चलो नेक्स्ट. Majority of insect pollinated flowers are large, colorful, fragrant and rich in nectar. ये लाइन कहीं मॉक टेस्ट में पूछी गई है, यहाँ पे चेंज करके कुछ भी पूछ सकते हैं, लार्ज की जगह स्मॉल, कुछ भी, बट करेक्ट है. Majority of insect pollinated flowers are large, colorful, fragrant and rich in nectar. When the flowers are small, a number of flowers are clustered into an inflorescence to make them conspicuous. Animals are attracted to flowers by color and or fragrance. The flowers pollinated by flies and beetles secrete fall odors to attract these animals. Important है ये टॉपिक, इसलिए मैं डबल बार पढ़ रहा हूँ लाइन. ठीक है? The flowers pollinated by flies and beetles secrete fall odors to attract these animals. Correct? To sustain animal visits, the flower have to provide rewards to the animals. Nectars and pollen grain are the useful floral rewards. कौन है? Nectar and pollen grain are the useful floral rewards. ठीक है? For harvesting the rewards from the flower, the animal visitor come in contact with the anthers and the stigma. इसके contact में आते हैं anther and stigma. The body of the animals gets a coating of pollen grain, which are generally sticky in animal pollinated flowers. When the animal carrying pollen on its body come in contact with the stigma, it brings about pollination. ठीक है. In some species, floral rewards are in providing safe places to lay eggs. An example is that of the tallest flower of Amorphophallus. The flower is itself about 6 feet in height. 6 feet की जगह सेंटीमीटर मीटर कुछ भी कर सकते हैं, बट करेक्ट है 6 feet. In some species, floral rewards are in providing safe places to lay eggs. An example is that of the tallest flower of Amorphophallus. The flower itself is about 6 feet in the height. A similar relationship exists between a species of moth and the plant yucca, where both the species moth and the plant cannot complete their life cycle without each other. The moth deposits its egg in the locule of the ovary, and the flower in turn gets pollinated by the moth. किससे pollinated होते हैं? moth से. ठीक है. The larva of the moth come out of the egg as the seeds start developing. The larva of the moth come out of the eggs as the seeds start developing. Clear? Why don't you observe some flowers of the following plants or any other available to you? Cucumber, mango, pipal, coriander, papaya, onion, lapia, lobia, cotton, tobacco, rose, lemon, eucalyptus, banana. Try to find out which animal visit them and whether they could be pollinated. You will have to patiently observe the flowers over a few days and at different times of the day. You could also try to see whether there is any correlation in the characteristics of a flower to the animal that visits it. Carefully observe if any of the visitors come in contact with the anther and the stigma, as only such visitor can bring about pollination. Many insects may consume pollen or the nectar without bringing about pollination. Such floral visitors are referred to as pollen nectar robbers. You may or may not be able to identify the pollinator, but you will surely enjoy your efforts. कोई काम का पैराग्राफ नहीं था ये, इसलिए मैंने स्पीड में पढ़ा. ठीक है. So don't try to be panic for that. Clear? Next is outbreeding devices. Majority of the flowering plants produce hermaphrodite flowers. Majority of the flowering plant produces hermaphrodite flowers. कौन से? Hermaphrodite flowers. And pollen grain are likely to come in contact with the stigma of the same flower. Continued self-pollination results in inbreeding depression. Continued self-pollination results in inbreeding depression. ठीक है. Flowering plants have been developed many devices to discourage self-pollination and to encourage cross-pollination. To discourage self-pollination and to encourage cross-pollination. ठीक है. In some species, pollen release and stigma receptivity are not synchronized. Either the pollen is released before the stigma becomes receptive, or a stigma becomes receptive much before the release of pollen. In some other species, the anther and the stigma are placed at different positions so that the pollen cannot come in contact with the stigma of the same flower. Both these devices prevent autogamy. The third device to prevent inbreeding is self-incompatibility. This is the genetic mechanism and prevents self-pollen from the same flower or the other flowers of the same plant from fertilizing the ovule by inhibiting pollen germination or pollen tube growth in the pistil. Another device to prevent self-pollination is the production of unisexual flowers. If both male and female flower are present on the same plant, such as castor and maize, that is monoecious. On a monoecious, castor and maize. If both male and female flower are present on the same plant, such as castor and maize, monoecious याद रख सकते हो, MC से. It prevents autogamy but not chitinogamy. Monoecious में क्या prevent करता है? Autogamy but not chitinogamy. In several species such as papaya, male and the female flower are present on the different plant. that is each plant is either male or female, that is dioicy. This condition prevents both autogamy and geitonogamy. This condition prevent both autogamy and geitonogamy, दोनों प्रिवेंट करता है. ठीक है, कौन सा वाला? डायोसी वाला कंडीशन. ठीक है, उसके एग्जाम्पल कौन है? पपाया. ठीक है. नेक्स्ट इज pollen pistil interaction. Pollination does not guarantee the transfer of the right type of pollination. Compatible pollen of the same species of the stigma. Often pollen of the wrong type, either from the other species or from the same plant, if it is self-incompatible, also land on the stigma. The pistil has the ability to recognize the pollen, whether it is of right type, that is compatible, or of the wrong type, that is incompatible. If it is of the right type, the pistil accepts the pollen. If it is of the right type, अगर सही टाइप का है तो पिस्टल एक्सेप्ट करेगी pollen and promotes post-pollination events. That leads to fertilization. यहाँ पर डायग्राम दिया हुआ है. ठीक है, एक डायग्राम दिया हुआ है pollen grain germinating on the stigma. दूसरा दिया हुआ है pollen tubes growing through the style. तीसरा दिया हुआ है LS pistil showing path of the pollen tube and large view of an egg apparatus दिया हुआ है showing entry of the pollen tube, discharge of the male gametes into synergy and the movement of the sperm, one into the egg and the other into the central cell का डायग्राम दिया हुआ है E वाला. ठीक है. So the next is, if the pollen is of the wrong type, the pistil rejects the pollen by preventing pollen germination on the stigma or the pollen tube growth in the style. Pollen tube की growth होती है style. The ability of the pistil to recognize the pollen followed by its acceptance or the rejection is the result of continuous dialogue between pollen grain and the pistil. Is the result of a continuous dialogue between pollen grain and the pistil. This dialogue is mediated by chemical components of the pollen interacting with those of the pistil. It is only in recent years that botanists have been able to identify some of the pollen and pistil components and the interaction leading to the recognition followed by acceptance or rejection. As mentioned earlier, following compatible pollination, the pollen grain germinate on the stigma to produce a pollen tube through one of the germpod. The content of the pollen grain move into the pollen tube. Pollen tube grows through the tissue of the stigma and style and reaches the ovary. You would recall that in some plants, pollen grain are shaded at two-celled condition, that is, a vegetative cell and a generative cell. वो होती है two-celled condition. In such plant, the generative cell divides and form the two male gamete during the growth of pollen tube in the stigma. ठीक है, कब होता है? Generative cell divides and form the two male gamete during the growth of pollen tube in the stigma. In plants which shed pollen in the three-celled condition, pollen tubes carry the two male gametes from the beginning. Clear? In plants which shed pollen in the three-celled condition, in plant which shed pollen in the three-celled condition, pollen tubes carry the two male gametes from the beginning. शुरू से ही two male gamete carry करते हैं. ठीक है. Pollen tube, after reaching the ovary, enters the ovule through the micropyle and then enters one of the synergid through the filiform apparatus. Pollen tube, after reaching the ovary, enters the ovule through the micropyle and then enters one of the synergid through the filiform apparatus. Many recent studies have shown that filiform apparatus present at the micropylar part of the synergid guides the entry of the pollen tube. Filiform apparatus कहाँ पे present होता है? At the micropylar part of the synergid guides the entry of the pollen tube. कौन करता है entry? Filiform apparatus. ठीक है. All these events from the pollen deposition on the stigma until the pollen tube enters the ovule are together referred to as pollen-pistil interaction. All these events from pollen deposition on the stigma until pollen tube enters the ovule are together referred to as pollen-pistil interaction. Clear? As pointed out earlier, pollen-pistil interaction is a dynamic process. कौन सी process है? Dynamic process involving pollen recognition followed by promotion or inhibition of the pollen. The knowledge gained in this area would help the plant breeder in manipulating pollen-pistil interaction, even in incompatible pollination to get desired hybrids. You can easily study pollen germination by testing some pollens from flowers such as pea, chickpea, crotalaria, balsam, and vinca on a glass slide containing a drop of sugar solution about 10% after about 15 to 30 minutes. Observe the slide under the low power lens of the microscope. You are likely to see the pollen tubes coming out of the pollen grain. ठीक है, ये लाइन काम की नहीं थी इसलिए स्पिन में पड़ी. As you shall learn in the chapter on plant breeding in chapter 9, a breeder is interested in crossing different species and often genera to combine desirable character to produce commercially superior varieties. Artificial hybridization is one of the major approaches of crop improvement program. In such crossing experiments, it is important to make sure that only the desired pollen grains are used for pollination and the stigma is protected from contamination, that is from unwanted pollen. This is achieved by emasculation and bagging technique. If the female parents bear bisexual flowers, removal of anther from the flower bud before the anther dehisces using a pair of forceps is necessary. If the female parent bears bisexual flowers, removal of anther from the flower bud before the anther dehisces using a pair of forceps is necessary. This step is referred to as emasculation. Clear? Emasculated flowers have to be covered with a bag of suitable size, generally made up of butter pepper to convert contamination of its stigma with unwanted pollen. Emasculated flowers have to be covered with a bag of suitable size, generally made up of butter pepper, to prevent contamination of its stigma and the unwanted pollen. This process is called bagging. When the stigma of bagged flower attains receptivity, mature pollen grains collected from anther of the male parent are dusted on the stigma. Mature pollen grain collected from anthers of the male parent are dusted on the stigma, and the flower are rebaked and the fruits allowed to develop. If the female parent produces unisexual flowers, there is no need for emasculation. कब नहीं होगी no need? If the female parent produces unisexual flower. The female flower buds are baked before the flowers open. When the stigma becomes receptive, pollination is carried out using the desired pollen and the flower is rebaked. Next we are going to read double fertilization. After entering one of the synergids, the pollen tube releases the two male gametes into the cytoplasm of the synergid. कहाँ पे रिलीज करती है? Into the cytoplasm of synergids. The pollen tube releases the two male gametes in the cytoplasm of the synergid. चेंज करके cytoplasm की जगह कुछ भी कर सकता है. करेक्ट है cytoplasm. One of the male gametes move towards the egg cell and fuses with its nucleus, thus completing the syngame. ठीक है, क्या complete होगी? syngame. This results in the formation of a diploid cell, the zygote. Zygote is a diploid cell. The other male gametes move towards the two polar nuclei located in the central cell and fuses with them to produce a triploid primary endosperm nucleus, that is PEN. As this involves the fusion of three haploid nuclei, it is termed as triple fusion. Since two types of fusion, that is syngamy and triple fusion takes place in an embryo sac, the phenomenon is termed as double fertilization, an event unique to flowering plants. The central cell after triple fusion becomes the primary endosperm cell and develops into the endosperm while the zygote develops into an embryo. Important line here. The central cell after triple fusion becomes the primary endosperm cell and develops into the endosperm while the zygote develops into an embryo. डायग्राम दिया हुआ है यहाँ पे फर्टिलाइज एम्ब्रियो सेक का शोइंग जाइगोट एंड प्राइमरी एंडोस्पर्म न्यूक्लियस. ठीक है, पीएन भी दिया हुआ है और प्राइमरी एंडोस्पर्म सेल भी दिया हुआ है. ठीक है, दोनों डिफरेंट चीज़ है. प्राइमरी एंडोस्पर्म न्यूक्लियस 3n है, जाइगोट 2n है. ठीक है, और बाकी यहाँ स्टेजेस दी हुई है एम्ब्रियो डेवलपमेंट इन ए डाइकोट शो इन रेड्यूस्ड साइजेस कंपेयर टू ए. सो द नेक्स्ट इज पोस्ट फर्टिलाइजेशन structures and events. Following double fertilization, events of endosperm and embryo development, maturation of ovule into seed and ovary into fruit are collectively termed post-fertilization events. Following double fertilization, events of the endosperm and embryo development, maturation of ovule into seed and ovary into fruit are collectively termed as post-fertilization events. में आता है events of endosperm and embryo development, मैचुरेशन ऑफ ओव्यूल इंटू सीड एंड ओवरी इंटू फ्रूट आते हैं पोस्ट फर्टिलाइजेशन इवेंट में. सो द नेक्स्ट इज एंडोस्पर्म. एंडोस्पर्म डेवलपमेंट प्रीसीड्स एम्ब्रियो डेवलपमेंट. एंडोस्पर्म डेवलपमेंट प्रीसीड्स एम्ब्रियो डेवलपमेंट. एंडोस्पर्म डेवलपमेंट प्रीसीड्स एम्ब्रियो डेवलपमेंट. ठीक है. वाई द प्राइमरी एंडोस्पर्म सेल डिवाइड्स रिपीटेडली and forms a triploid endosperm tissue. The cells of this tissue are filled with reserve food materials and are used for the nutrition of the developing embryo. In the most common types of endosperm development, the primary endosperm nucleus undergoes successive nuclear division to give rise to free nuclei. This stage of endosperm development is called as free nuclear endosperm. In most common type of endosperm development, the primary endosperm nucleus undergoes successive nuclear division to give rise to free nuclei. This stages of endosperm development is called free nuclear endosperm. Subsequently, cell wall formation occurs and the endosperm become cellular. The number of free nuclei formed before cellularization varies greatly. The coconut water from tender coconut that you are familiar with is nothing but free nuclear endosperm made up of thousands of nuclei, and the surrounding white kernel is the cellular endosperm. जो आसपास की सराउंडिंग होती है white kernel is the cellular endosperm. वो क्या होती है? cellular endosperm होती है. ठीक है. Endosperm may either be completely consumed by the developing embryo. Example, pea, groundnut, beans. इसको ऐसे याद रख सकते हैं, पेगू बी, पीग्रो बी से नाम रख सकते हैं याद. पी से पी, ग्रो से ग्राउंडनट, बी से बीन्स. एंडोस्पर्म में either be completely consumed by the developing embryo. Example, pea, groundnut, beans. Before seeds maturation, or it may persist in the mature seeds. Example, custard and coconut. इनमें क्या होता है? persist होता है the mature seed. उसके example है custard and coconut. ठीक है. and be used up during seed germination. Again I am repeating the line. Endosperm may either be completely consumed by the developing embryo. Example, pea, groundnut, beans. Before seed maturation, or it may persist in the mature seed, custard and coconut, and be used up during seed germination. Clear? Split open some seeds of custard, pea, beans, groundnut, fruits of coconut, and look for the endosperm in each cases. Find out whether the endosperm is persistent in cereals, wheat, rice, and maize. करो ये काम. ठीक है. डायग्राम बताऊं कि इधर जो दिया हुआ है एक डायग्राम टिपिकल डाइकॉट एम्ब्रियो का दिया हुआ है और एलएस ऑफ एन एम्ब्रियो ग्रास का दिया हुआ है। रेडिकल दिया हुआ है इसमें नीचे रूट केप कॉलराइजा ब्लास्ट शूट अपेक्स कोलेप्टाइल कार्टिलेजन हाइपोकॉटाइल रेडिकल रूट कैप प्लूमल। सो द नेक्स्ट इज एम्ब्रियो। क्या पढ़ने वाले हम? एम्ब्रियो। एम्ब्रियो डेवलप्स एट द माइक्रोपाइलर एंड ऑफ द एम्ब्रियो sac where the zygote is situated. Embryo develops at the micropylar end. कौन से वाले end? Micropylar end of the embryo sac where the zygote is situated. Most zygotes divide only after certain amount of endosperm is formed. Most zygotes divide only after certain amounts of endosperm is formed. जब certain amount of endosperm formed होगा तभी most of the zygote divide होंगे, ठीक है। This is an adaptation to provide assured nutrition to the developing embryo. Though the seeds differ greatly, the early stages of the embryo development, that is embryogeny, are similar in both monocotyledons and dicotyledons. Figure depicts the stages of embryogeny in a dicotyledonous embryo. वो दिया हुआ ऊपर डायग्राम। The zygote give rise to the proembryo and subsequently to the globular, heart-shaped and mature embryo. The zygote give rise to the proembryo and subsequently to the globular, heart-shaped and mature embryo. A typical dicotyledonous embryo consists of an embryonal axis and two cotyledons. Consists of an embryonal axis and the two cotyledons. The portion of embryonal axis above the level of the cotyledons is the epicotyle, which terminates with the plumule or stem tip. Plumule or stem tip. The portion of embryonal axis above the level of cotyledon is epicotyle, which terminates with the plumule or stem tip. The cylindrical portion below the level of cotyledon is hypocotyle. नीचे है मतलब hypocotyle. That terminate at its lower end in the radical or root tip. ठीक है। एपिकोटाइल में आता है प्लूमल और स्टेम टिप और हाइपोकोटाइल में आता है लोअर एंड टर्मिनेट, ठीक है। हाइपोकोटाइल में आता है दैट टर्मिनेट एट लोअर एंड इन द रेडिकल और रूट टिप। हाइपोकोटाइल में रेडिकल और रूट टिप। द रूट टिप इज कवर्ड विथ ए रूट कैप। रूट की टिप होती है कवर्ड होती है रूट कैप से, ठीक है। क्लियर है। एपिकोटाइल में टर्मिनेट विद द प्लूमल और स्टेम टिप एंड हाइपोकोटाइल टर्मिनेट एट इट्स लोअर एंड हाइपोकोटाइल दैट टर्मिनेट एट इट्स लोअर एंड इन द रेडिकल और द रूट टिप, ठीक है। क्लियर। एम्ब्रियोस ऑफ मोनोकोटलीडेंस पोसेसेस ओनली वन कोटलीडन। बोलने की बात है क्या ये भी? इन द ग्रास फैमिली द कोटलीडन इज कॉल्ड सूटेलम that is situated towards the one side lateral of the embryonal axis. That is situated towards one side. कौन सी side? That is situated towards one side. कौन सी? lateral of the embryonal axis. ठीक है, clear? At its lower end, the embryonal axis has the radical and root cap. At its lower end, the embryonal axis has the radical and root cap enclosed in an undifferentiated sheath called coleorhiza. Lower end, the embryonal axis is the radical and root cap. Lower end पे क्या होती है? Lower end at its lower end की embryonal axis has the radical and the root cap enclosed in an undifferentiated sheath called the coleorhiza. The portion of the embryonal axis above the level of attachment of scutellum is the epicotyle. Epicotyle has a shoot apex and a few leaf primordia enclosed in a hollow foliar structure, the coleoptile. Epicotyle has a shoot apex and a few leaf primordia enclosed in a hollow foliar structure, the coleoptile. ठीक है। एपिकोटाइल enclosed indulged in a holofollial structure, the coloptile. Soak a few seeds in water, say of wheat, maize, peas, chickpeas, groundnut, overnight. Then split the seeds and observe the various part of the embryo and the seed. करो ये काम, ठीक है? कोई काम का नहीं है वैसे तो, ठीक है? So the next is seed. So let's proceed further with the seed. In angiosperm, the seed is the final product of the sexual reproduction. कौन है final product? Angiosperm में seed is the final product of sexual reproduction. It is often described as a fertilized ovule. It is often described as fertilized ovule. Seeds are formed inside the fruits. इनके अंदर fruits के अंदर seeds are formed inside the fruits. A seed typically consists of seed coat, cotyledons, and an embryo axis. A seed typically consists of seed coat, cotyledon, and an embryo axis. ठीक है, तीन चीजों से seed coat, cotyledon, and embryo axis. The cotyledon of the embryo are simple structures, generally thick and swollen due to storage of food reserve, as in legumes. The cotyledon of the embryo are simple structures, generally thick की जगह thin कर सकते हैं, but correct है generally thick, and swollen due to the storage of food reserve, that is as in legumes. Mature seed may be non-albuminous or X-albuminous. Non-albuminous seeds have no residual endosperm and this is completely consumed during embryo development. Non-albuminous seeds में no residual होता है endosperm का, it is completely consumed during embryo development. जिसका एग्जाम्पल है pea एंड ग्राउंडनट. पी और ग्राउंडनट है एग्जाम्पल नॉन एल्ब्यूमिनस सीड्स का, ठीक है? क्लियर? पी एंड ग्राउंडनट आर द एग्जाम्पल ऑफ नॉन एल्ब्यूमिनस सीड्स. Albuminous seeds retain a part of the endosperm as it is not completely used up during embryo development. पूरा यूज नहीं होता है, इसलिए seed retain a part of the endosperm as it is not completely used up during embryo development. Example है wheat, maize, barley, custard. Albuminous seeds के example है wheat, maize, barley and custard. Wheat, maize, barley and custard. एग्जाम्पल है एल्ब्यूमिनस सीड के, ठीक है? एल्ब्यूमिनस के एग्जाम्पल है BMWC. B से बार्ली, M से मैज, W से व्हीट, C से कस्टर्ड. क्लियर? माइंड में सेट. Occasionally in some seeds such as black pepper and beet, remnants of nucellus are also persistent. Remnant of nucellus किसमें होते हैं? persistent. In some seeds such as black pepper and beet. In some seeds such as black pepper and beet, remnants of nucellus are also persistent. This residual persistent nucellus is the perisperm. This residual persistent nucleus is the perisperm. क्या है? residual persistent nucleus is the perisperm. Integuments of ovules harden as a tough protective seed coat. The micropyle remain as a small pore in the seed coat. The micropyle remain as a small pore in the seed coat. This facilitate entry of oxygen and water into the seed during germination. कौन? micropyle, ठीक है? As the seed matures, its water content is reduced and seeds become relatively dry, that is 10 to 15% moisture by mass. 10 to 15% moisture by mass. The general metabolic activity of the embryo slows down. The embryo may enter a state of inactivity called dormancy, or if favorable conditions are available, that is adequate moisture, oxygen and suitable temperature, they germinate. Favorable condition है adequate moisture, oxygen and suitable temperature, तो क्या करेगी? germinate. As ovule mature into the seed, the ovary develop into a fruit. That is, the transformation of ovule into seed and ovary into fruit proceeds simultaneously. ठीक है? simultaneously means साथ साथ होता है. The wall of the ovary develop into the wall of the fruit called pericarp. The wall of the ovary develops into the wall of fruit called pericarp. ठीक है, क्या होती है? The wall of the ovary develops into the wall of fruit called pericarp. ठीक है, the fruits may be fleshy as in guava, orange. The fruit may be fleshy as in guava, orange, mango, etc. Fleshy के example है guava, mango, etc. Guava, mango, orange, etc. और may be dry as in groundnut and mustard. Dry के example है mustard और groundnut. ठीक है, mustard and groundnut example है dry के. Many fruits have evolved mechanism for dispersal of seeds. Many fruits have evolved mechanism for dispersal of seeds. Recall the classification of fruits and their dispersal mechanism that you have studied in an earlier class. Is there any relationship between number of ovules in an ovary and the number of seed present in a fruit? है क्या कोई relationship? बताओ. Is there any relationship between number of ovules in an ovary and the number of seeds present in a fruit? Yes, there is a relationship between the number of ovules in the ovary and the number of seeds in a fruit. That is, if all the ovules in an ovary are fertilized, then the number of seeds in the fruit will be the same as the number of ovules. ठीक है, clear? In most plants, by the time the fruit develops from the ovary, other floral parts degenerates and fall off. However, in a few species such as apple, strawberry, cashew, etc., the thalamus also contribute to fruit formation. कौन में? एप्पल, स्ट्रॉबेरी और कैशू में. एप्पल, स्ट्रॉबेरी और कैशू में थैलामस आल्सो कंट्रीब्यूट टू फ्रूट फॉर्मेशन. फ्रूट की जगह सीड फॉर्मेशन कर देगा तो रॉन्ग स्टेटमेंट हो जाएगा. ठीक है, सच फ्रूट्स आर कॉल्ड फॉल्स फ्रूट. फॉल्स फ्रूट के एग्जाम्पल है स्ट्रॉबेरी, कैशू और एप्पल. एप्पल, स्ट्रॉबेरी, कैशू. मोस्ट फ्रूट्स हाउएवर डेवलप ओनली फ्रॉम द ovary and are called the true fruits. Although in most of the species, fruits are the result of fertilization, there are a few species in which fruits develop without fertilization. Fruit develop without fertilization. Such fruits are called parthenocarpic fruits. Banana is one such example of the parthenocarpic fruit. Parthenocarpy can be induced through the application of the growth hormone, and such fruits are seedless. अब यहाँ पर डायग्राम दिया हुआ है कॉटलीडन्स का, माइक्रोपाइल का. ठीक है, सीड दी हुई है. ठीक है, सीड कॉट, एंडोस्पर्म दिया हुआ है. ठीक है, कॉटलीडन, हाइपोकॉटाइल शूट, सीड कॉट, कॉटलीडन दिया हुआ है, शूट, एपिकल मेरिस्टेम, हाइपोकॉटाइल रूट एक्सिस, रूट कैप, एंडोस्पर्म, पेरिकार्प, एंडोस्पर्म, स्विचेलम, कोलेप्टाइल, प्लुमेल, रेडिकल राइजा, थैलामस दिया हुआ है, सीड दी हुई है, एंडोकार्प, मेसोकार्प, थैलामस. एक इनका डायग्राम दिया हुआ है. फॉल्स फ्रूट ऑफ एप्पल एंड स्ट्रॉबेरी के डायग्राम दिए हुए हैं. ठीक है, सीड ऑफर सेवरल एडवांटेजेस टू द एंजियोस्पर्म्स. फर्स्टली, since reproductive process such as pollination and fertilization are independent of water, seed formation is more dependable. Since reproductive process such as pollination and fertilization are independent of water, since reproductive processes such as pollination and fertilization are independent of water, seed formation is more dependable. Also, seeds have better adaptive strategies for disposal to new habits, and help the species to colonize in the other area. As they have sufficient food reserve, young seedlings are nourished until they are capable of photosynthesis on their own. The hard seed coat provides protection to the young embryo. Being products of sexual reproduction, they generate new genetic combination leading to the variation. Seed is the basis of our agriculture. Dehydration and dormancy of mature seeds are crucial for storage of seeds, which can be used as food throughout the year and also to raise crop in the next season. Dehydration and dormancy of mature seeds are the crucial for storage of seeds, which can be used as food throughout the year and also to raise crop in the next season. Can you imagine agriculture in the absence of seeds or in the presence of seeds which germinate straight away soon after the formation and cannot be stored? How long do the seeds remain alive after they are dispersed? This period again varies greatly. In a few species, the seed loses viability within a few months. Seeds of a large number of species live for several years. Some seeds can remain alive for hundreds of years. There are several records of very old yet viable seeds. The oldest is that of lupin, that is Lupinus arcticus, excavated from the Arctic tundra. The oldest is that of lupin, that is Lupinus arcticus, excavated from Arctic tundra. The seed germinated and flowered after an estimated record of 10,000 years of dormancy. The seeds germinated and flowered after an estimated record of 10,000 years of dormancy. A recent record of 2,000-year-old viable seed is of the date palm. 2,000-year-old viable seed is of the date palm. Phoenix dactylifera discovered during the archaeological excavation of the King Herod's palace near the Dead Sea. Date palm की थी जिसका नाम था Phoenix dactylifera, discovered during the archaeological excavation of King Herod's palace near the Dead Sea. 2000-year-old, ठीक है. After completing a brief account of sexual reproduction of flowering plant, it would be worth attempting to confront the enormous reproductive capacity of some flowering plant by asking the following questions. How many eggs are present in an embryo sac? How many embryos are present in an ovule? How many ovules are present in an ovary? How many ovaries are present in a typical flower? How many flowers are present on a tree? And so on. Can you think of some plants in which fruit can contain very large number of seeds? Orchid fruits are one such category, and each fruit contain thousands of tiny seeds. Okay, orchid contain a very large number of seeds. Similar is the case of fruits. Similar is the case in fruits of some parasites. Similar is the case in fruits of some parasitic species such as Orobanchian striga. Have you seen a tiny seed of ficus? How large is the tree of ficus developed from the head of the tiny seed? How many billions of seeds does each ficus tree produce? Can you imagine any other example in which such a tiny structure can produce such a large biomass over the years? Can you? So, what we are going to read is apomixis and polyembryony. चलो तो वो क्वेश्चन के आंसर बताते हैं, ठीक है. हाउ मेनी एग्स आर प्रेजेंट इन एन एम्ब्रियो सेक? An embryo sac contains one egg, also known as female gamete. Clear? How many embryo sacs are present in an ovule? An ovule generally has one embryo sac. ठीक है, clear? How many ovules are present in an ovary? The number of ovules in an ovary depends on the flower and can range from one to many. Understood? Clear? How many ovaries are present in a typical flower? A typical flower has one ovary, but the number of ovary can vary depending on the flower. Okay? Understood? So let's proceed with the apomixis and polyembryony. Although seeds in general are the products of fertilization, a few flowering plants such as some species of Asteraceae and grasses, Asteraceae and grasses, have evolved a special mechanism to produce seed without fertilization called apomixis. Seed without fertilization is called apomixis. What is the fruit production without fertilization called? Fruit production without fertilization is called parthenocarpy. Am I right? Yes, you are absolutely right. सही बोला. Thus, apomixis is a form of asexual reproduction that mimics sexual reproduction. मजाक उड़ा रहा है भाई. Sexual reproduction. Thus, apomixis is a form of asexual reproduction that mimics sexual reproduction. There are several ways of development of apomictic seeds. In some species, the diploid egg cell is formed without reduction division and develops into the embryo without fertilization. Clear? Understood? There are several ways of development of apomictic seeds. In some species, the diploid egg cell is formed without reduction division and develops into the embryo without fertilization. Clear? More often, as in many citrus and mango varieties, some of the new cellular cells surrounding the embryo sac starts dividing, protrude into an embryo sac and develop into the embryo. In such species, each ovule contains many embryo. Occurrence of more than one embryo in a seed is referred to as polyembryony. जिसके example is citrus and mango. More often, as in many citrus and mango varieties, some of the new cells surrounding the embryo sac start dividing, that is protrude into the embryo sac and develop into the embryo. In such species, each ovule contains many embryo. Occurrence of more than one embryo in seed is referred to as polyembryony. Take out some seeds of orange and squeeze them. Observe the many embryo of different sizes and shapes from each seed. Count the number of embryos in each seed. What would be the genetic nature of the apomictic embryo? Can they be called cones? Can they be called cones? क्यों जी? Hybrids, variety of several of our food and vegetable crops, are being extensively cultivated. What would be the genetic nature of apomictic embryo? Apomictic embryo are genetically identical to their parent and are considered clones. ठीक है? So, yes, apomictic embryos are genetically identical to their parent and can be called clones. ठीक है? Hybrid variety of several of our food and vegetable crops are being extensively cultivated. Cultivation of hybrids has tremendously increased productivity. One of the problems of the hybrid is that seeds have to be produced every year. If the seeds collected from hybrids are sown, the plant in the progeny will segregate and do not maintain hybrid character. Production of hybrid seed is costly and hence the cost of hybrid seeds become too expensive for the farmer. If the seed collected from hybrid are sown, the plant in the progeny will segregate and do not maintain hybrid character. If these hybrids are made into apomix, there is no segregation of character in the hybrid progeny. Then the farmer can keep on using the hybrid seeds to raise new crop year after year, and he does not have to buy hybrid seeds every year. ठीक है? Thus, then the farmer can keep on using the hybrid seeds to raise new crop year after year and does not have to buy hybrid seeds every year. Because of the importance of the apomixis in hybrid seed industry, active research is going on in many laboratories around the world to understand the genetic of apomixis and to transfer apomictic genes into the hybrid varieties. Next is summary. We are going to read. Flower are the seat of sexual reproduction in angiosperm. In the flower, androecium consisting of stamens represent the male reproductive organ. Gynoecium consisting of pistils. GC represents a female reproductive organ. Atypical anther is bilobed, dithecous and tetrasporangate. Bilobed, dithecous and tetrasporangate. Pollen grain develop inside the microsporangia. Four wall layers, the epidermis, endothecium, middle layer and the tapetum surround the microsporangium. Cells of the sporogenous tissue lying in the center of the microsporangium undergo meiosis, that is microsporogenesis, to form tetrads of microspore. Individual microspore mature into the pollen grain. Pollen grain represent the male gametophytic generation. pollen grain have a two-layered wall, the outer exine and the inner intine. The exine is made up of sporopollenin and has a germ pore. Pollen grain may have two cells, a vegetative and generative cell, or three, a vegetative cell and two male gametes. At the time of shedding, the pistil has three parts: stigma, style, and ovary. Ovules are present in the ovary. The ovules have a stalk called funicle, protective stegment, protective integument. The ovule have a stalk called funicle, protective integuments, and an opening called micropyle. The central tissue is the nucellus, in which the archisporium differentiate. A cell of the archisporium, the megaspore mother cell, divides meiotically, and one of the megaspore forms the embryo sac, the female gametophyte. The mature embryo sac is 7-celled and 8-nucleate. At the micropylar end is the egg apparatus, consisting of two synergids and an egg cell. Micropylar end is the egg apparatus. At the micropylar end पे होता है egg apparatus, consisting of two synergid and the egg cell. At the chaliger end are the antipodal. At the center is a large central cell with two polar nuclei. Pollination is the mechanism to transfer pollen grain from the anther to the stigma. Pollinating agent are either biotic or abiotic. Abiotic का example है wind and water, and biotic के हैं animals. Abiotic का wind and water, biotic के animal. Pollen pistil interaction involves all events from the landing of the pollen grain on the stigma until the pollen tube enters the embryo sac. ठीक है? When the pollen is compatible, or pollen inhibition when the pollen is incompatible. Following compatible pollination, pollen grain germinates on the stigma and resulting pollen tube grows through the style and enters the ovule and finally discharges two male gametes in one of the synergids. Angiosperm exhibit double fertilization because of two fusion events occur in each embryo sac, namely syngamy and triple fusion. The products of this fusion are the diploid zygote and the triploid primary endosperm nucleus in the primary endosperm cell. Zygote develop into an embryo. And the primary endosperm cell (PEC) forms the endosperm tissue. Formation of endosperm always precedes development of the embryo. आ सकती है लाइन कहीं भी, ठीक है? Formation of the endosperm always आगे इसलिए important, always precedes development of the embryo. The developing embryo passes through different stages such as proembryo, globular, and heart-shaped stages before maturation. Mature dicotyledonous embryo has two cotyledons and an embryo axis with epicotyle and hypocotyle. Embryos of the monocotyledons have a single cotyledon. After fertilization, ovary develop into fruit and ovule develop into seed. A phenomenon called apomixis is found in some angiosperm, particularly in grasses. Phenomenon apomixis is found in this. It results in the formation of seed without the fertilization. Apomixes have several advantages in horticulture and agriculture. Some angiosperm produce more than one embryo in their seed. This phenomenon is called polyembryony. With this, we completed the chapter. रात के बजे हैं 1:20. So, thank you for hearing.